hope you enjoyed your lunch, and we're continuing to the second part of the day. And our next speaker is Professor David Donoho from Stanford University. He will speak about optimal shrinkage of singular values and eigenvalues in light of the spiked model.
Yeah, the title is, it should just be Principal Components Analysis in Light of the Spiked Model. It's not as scary as it sounds. And I'll try to explain what all the words mean as we go along. I first of all think it's great that the Feder family is willing to honor the fantastic work being done by the next generation. And been very impressed by the talk that we've heard so far. And looking forward to the further talks by the other awardees later today. Also like to say that I'm a statistician rather than, perhaps I should move this. I'm a statistician rather than an information theorist, but I am extremely grateful to the information theory community for being willing to publish some of my papers from time to time. And in fact, some of my most successful papers, but papers that no statistics journal would have touched at the time. So I think you're a very intellectually adventurous community, and I have utmost respect. I'm very lucky at Stanford that we have the electrical engineering department about 100 yards away, and the best place for coffee on campus. And so it's very easy to interact with them, and I've had great interactions with their department. We're also lucky that we have in the statistics department had joint appointments with electrical engineering over the years. In the past, Tom Cover, and currently we have Amir Dembo and Andrea Montanari, and it's just a fantastic thing to have those interactions. I wish there were more interactions between statistics and electrical engineering worldwide, because every time I've seen an interaction happening, it's been very fruitful for statistics anyway. Is there something I should do about the feedback? Could I make it better in some way?
Maybe you could lower maybe the volume or something there. There's like a volume button or something.
I have no idea.
I have no idea as well.
It's satisfactory.
Yeah, that's not clear. Yeah, I don't know. If you could do without it as well, maybe if people can hear without this, you can try.
I think that they need. Oops, that turned it up.
Yeah.
I think the point is they need it for recording or something.
So this one is not for recording. So maybe you can turn this one off and take it off. The other one is for recording. This is turned off. We can take this off. Yeah, the other one,
I don't know.
This one I think is. Ooh, we got mixed up here.
Okay. Anyway, as you can see, as you can see, there
the interactions between statisticians and engineers.
A number of related papers, which I'll try to cover some of the territory of these. I just don't know how far I'll get because this should be a general talk. So my longtime collaborator Ian Johnstone, also a professor at Stanford, Matan Gavish, who just got his PhD at Stanford, is now junior faculty at the Hebrew University, and a current PhD student at Stanford, Edgar Dobribon. And there's some papers that we've published or that are in archive and in process of going the rounds. So first of all, principal components analysis, we could say it's a ubiquitous process in data processing and data analysis throughout science and technology. And the basic schema is you have a data matrix, a rectangular array of numbers, and you either do a singular value decomposition of the matrix itself, or you do an eigenvalue decomposition of y prime y or y prime, depending on the format. Then you make a kind of plot of the eigenvalues or singular values, as the case may be, and then there are some big ones and there are some others, and the big ones you pay attention to and the other ones you ignore, and you make a story about it or some sort of application. Now, this is important, for example, in signal processing. You see it in radio frequency signal processing associated with things having to do with detecting signals incident on arrays. Here's just a generic paper applying techniques like this. It's in applied physics, and there's something about a sensor that's being set up, and then there's some data analysis, which I'll lay out as follows. There's an underlying data matrix which is connected at an array of sensors, and then when you do the singular value decomposition, you get a combination of, now how do I, you get first of all some singular values which are plotted here on their side, some singular vectors which are plotted in a false color on their side like this, and there's some big singular values and there's some small ones and there's some singular vectors, and a story is made about which singular values are important, which singular vectors are interpretable, and how that indicates that this sensor is appropriately able to localize some signals. If we go to computational biology, we can come to an example of a paper. It goes back to a style of reasoning attributed to Luca Cavalli-Sforza, that you can take genomic data and you can recover the basic residence patterns of people going back centuries. So in this particular paper from a group of, well, as it turns out, statisticians, computational biologists, and others, there's an array of SNP data, and so that's a rectangular data set, which is, let's say it's in the 100 Ks going this way, and maybe something like 1,000 to 3,000 data points going this way. These are individuals, and going this way is genomic data that corresponds genomic measurements. And so for each individual, we have a row, and at various sites along that row, we have numbers that are 0, 1, or 2 that have something to do with whether you have the dominant allele in that position or a variant allele. And you can take a matrix of this kind, you can do the singular value decomposition of that matrix, and what you'll see is that you have some, or you can do the eigenanalysis of the corresponding quadratic form that associated with the matrix, as I indicated earlier, and you'll get some eigenvalues. In this case, what you see in these data, they're taken from individuals all around Europe, and what you'll see is there are 2 large eigenvalues, and then there's a whole bunch down here in what's called the Scree plot that don't seem to be very distinctive. There's one that happens to be mathematically 0 because a mean subtraction was done somewhere in the processing. It forces a 0 eigenvalue. We ignore that. And now, if you look at the eigenvectors that come out, those are vectors going down this way. So there are as many coordinates in an eigenvector as there are people. And you can view that as giving you a number per person. If you take the first 2, you get 2 numbers per person. And you can use that to plot the individuals. And as you see here, this plot reveals, so that there's one individual there, and there's another individual there, et cetera. They're being plotted on some axes, which are called PC1 and PC2, associated with those eigenvalues. And what you see is you've recovered a map of Europe. Namely, the individuals who went to make up this study were not, the perpetual wandering Jews. I think these were rooted people who had been for generations in place. And you can see from this that the data sort themselves out. And of course, there's a map of Europe. You can ask, well, you actually know where these people live, and you can ask if you've recovered those places, and there's an average error of at most 500 kilometers in placing the people, and we can say more and so on and so forth. But the first two eigenvectors have located the people, and the first two eigenvalues stick out and are calling attention to themselves. And so this methodology obviously has some significance and utility. This is an example of principal component analysis. It's widely practiced in science and technology, as we've said. In fact, Uli Maurer is here, so we've got Swiss people. I can point out that if you take the Swiss data in here, just the individuals from Switzerland, and then just make a plot zooming in on that you even recover the linguistic separation in that country. So that actually, if we color the individuals by their particular native language, you'll see that the French are recovered by this technique to be in a different place than the Germans and so on. So there's some real signal about something that we know to be truth that's emerging from the analysis of what otherwise seems like extremely noisy data, simply because there are genetic events happening, at the time of meiosis, which are driving this, and they're really like tossing a coin. Nevertheless, with all of that noise that's present from these genomic interactions, we're able to recover something as fundamental as the rootedness patterns of the human civilization over time. Now, there's an important variation, which is that we do the exact opposite. Namely, instead of paying attention to the big eigenvalues, we ignore them and we look at the part that's explained by everything else. And you see this also in the studies of so-called copy number variation, which are also from genomics. And this is an article from the Eichler lab. There, in a similar way, you have data which is in the same format and of similar extent, hundreds of thousands by thousands, but the actual numbers in here are a little different. So instead of being 0, 1, and 2, you could maybe have a 3 or a 4, usually 0, 1, or 2, etc. It's a copy number variation. It's a different meaning. And it turns out there that the top principal components are artifacts that have to do something often with what's being measured, the measuring device, and you want to take them out and look at what's left. So here they show what the raw data look like. The individual rows are plotted out as signals. Then you take the singular value decomposition, you remove the biggest contributions, and then reconstruct. Now you have something much less noisy, and some genomic signal can pop up above it. So you see both patterns, and that's a tribute to the fertility of this whole method, that it can be used in a variety of different ways. So it's difficult to capture it all in just one way, but you have to know that it's a ubiquitous technology. In looking at the data associated with this particular example, a plot of the eigenvalues will show you that there's, again, a few big ones, and these are the things that correspond to the nuisance that you want to remove rather than the signal that you want to find. Now, this plot that we've been making, for example, here, where you have eigenvalue or singular value versus order, is an obvious enough thing to do, but someone first wrote a paper about it, and that was Cattell in 1966, so it's got its 50-year birthday today. Just in Google citations, it has well over 8,000, and every year many more. This is a very heavily practiced method in science and technology. You're trying to say there's some signal buried in this array, and you look for something to pop out and vindicate your assertion. So the original article indeed had the kind of plot we're talking about, and there was this notion present in that original article that what you typically saw was somehow eigenvalues that are moving along a straight line, and then boom, something breaks, and the last few are much bigger. And we saw exactly this logic earlier in the applied physics example, and we saw this logic in the genes from the geography from genes example, and we saw this logic being used in the copy number variation, and there's endless examples of this kind of thing. Now, of course, what we knew 50 years ago was much less than what we know today about the properties of these techniques, so maybe we can give a little more basis for this set of ideas. So let's bring in some random matrix theory. Let's imagine that we have an array that's IID Gaussian. Let's imagine that actually we have a sequence of such arrays growing to increasing sizes. As you can see, that's relevant because in the applications we really do look at pretty big arrays. And let's have a defined asymptotic shape for these things. And then let's consider either doing a singular value decomposition of the array or an eigen decomposition. And of course, those are connected ideas in such a way that if we set everything up right, then maybe the singular values of one when squared will be the eigenvalues of the other. And I just sort of operate interchangeably without talking much about this. I think it's an understood idea. Okay, so the first thing that we now understand about this that maybe wasn't understood years ago is that if you have a large matrix which is mostly noise, then there's a defined bulk distribution which just depends on the asymptotic shape of the matrix. You know, is it square? Is it wide? Is it tall? And the parameter beta that controls that controls the shape. It controls also the shape of the bulk distribution of the eigenvalues. So this is called the Marchenko-Pastur distribution. And for example, if I have a square, then I'm at beta equals 1, as many rows as columns, and I have this density, whereas if I go to lower betas, it becomes more tall, and I have that the density becomes more concentrated, about 1, and actually asymptotically becomes something that's kind of like a parabola, very narrow, around 1. And I could go and generate a big random matrix empirically with 1,000 by 1,000 size and get the eigenvalues. Well, what I mean by that is, you know, take z prime z and get their eigenvalues. Then I'll get a histogram like this. The Marchenko-Pastur prescribes the beta equals 1 shape, and this is what you'll see is that there's a reasonable match, and we could drill down into it, and you would see if we did statistical testing of a very, you know, careful nature, that it would line up very accurately. Now, if we talk about singular values, well, because they're the square roots, we already sort of know what the distribution has to be. It's induced by the Marchenko-Pastur. But one thing that's of interest is there's a specific case that is nice to know about, which is that if it's a square matrix and you look at the singular values, they are going to come from the so-called quarter circle law, namely the histogram, when appropriately scaled, fills out a perfect quarter circle. And now this is not quite appropriately scaled. This is beta equals 1, the square case for singular values. And we would have to, you know, make this height the same as this width, and then you would see a quarter circle. So here's an approximate example where we got the singular values of 1,000 by 1,000 square matrix, and you can see this is what happens for the bulk distribution. Now, what's the spiked model in the title? So what we were talking until now, so here's some MATLAB. Ignore this part for the moment, the part before the plus. What we were talking before on the previous slide is if I took a random matrix that you know, just consisted of noise, then we get the quarter circle law. But now if I have something that adds in some additional structure on top of that, then I have an example from the spiked model. And in this particular case, what you see is that the Scree plot of the singular values has pretty much linear shape, but then two things sticking out. Or if we went to the histogram, you have a quarter circle and then two things sticking out. The one thing I want to point out to you, which is important, is that these numbers here are not those numbers there. So there's some systematic displacement of the singular values away from what the true underlying numbers you would think ought to be. And so what you witness in the singular value decomposition of the empirical data is not a literal rendition of what actually generated it. It's not 100% faithful, and that's important for our story. There's a distortion here going on. The spiked model nomenclature was introduced by Ian Johnstone, and in American vernacular anyway, spiked punch is where you take some, fruit drink or something or other and you pour vodka in it. Okay, so before we were talking about noise and the bulk distribution that's present with noise. So that's not a very interesting pursuit, although probabilists have been happy to go there for years and years. But we wanted to put something a little bit more rewarding in it, and so we spike it. And that's the little bit of signal that we're talking about. So here, there's the kind of noise matrices we were talking about before. Then there's a low rank structure that's put inside it. There's an asymptotic shape. And the low rank structure is put in such a way that there are fixed singular values independent of the problem size, padded out by zeros for the signal. So there's a fixed low rank structure with fixed singular values, and we're allowed to make them fixed by the normalization we've been using on the noise, where the columns have unit norm. Although the model in covariance analysis has been around for quite a while, the use of this in connection with this particular structure is a bit more recent. It's also been called signal plus noise matrices, for example, by Jack Silverstein. And this is a very simple model, I think you'll agree, but you can see all the things that we're talking about. So the first thing that happens when we have this is that under the kind of asymptotics we just figured out, instead of all the singular values tending to the bulk distribution, it's just the singular values beyond the rth one. So there's R that you've spiked, and then the other ones tend to the bulk distribution and are going to have the kind of generalized quarter circle distribution. But the first couple are going to tend to their own limit, which is dependent on the signal amplitude but is not identical to the signal amplitude. There's a kind of specific asymptotic bias. So getting more and more data doesn't make the singular values that you observe more accurate reflections of the underlying signal strength. It just locks in the bias stronger and stronger so that you can't really be confused. It's really, really present. So if you wanted to, you could, we knew that the numbers that are associated with the underlying signal here were actually 1.7 and 2.3, but we're seeing something that's more like 2.5 and almost 3 coming out, and we could use this formula and put in beta equals 1, and we would see exactly that this formula is predicting what we in fact see on this plot. Now, even more interestingly, you can get that the singular vectors, which as we saw, are of some fundamental interest. For example, in recovering the rootedness patterns in Europe, well, we can talk about how accurate those are in recovering the true underlying signal vector directions. And in fact, we can get the cosine squared from either the right or left singular vectors according to some fixed expressions that are going to involve the shape factor of the matrix in some way. And there's a variety of literature over the years, starting with Bake, Benarousse, and Péchet on the spike model. The specific papers that we rely on a lot are maybe the most recent ones. There's also work by Boaz Nadler, which actually I find the easiest to understand, which is in Annals of Statistics 2007. Many, many papers over the period of time that this covers. So if we go back to the Scree plot and ask, what does the spike model say about it? So now it makes perfect sense that if there's a true underlying low-rank structure, which is namely, these genes are essentially, the genetic makeup is essentially determined by geography. There's a rank 2 structure because the surface of the Earth is two-dimensional and because human populations have been rooted for hundreds of years in the same place. And so the gene makeup of individuals there distributed across hundreds of thousands of SNPs is very indicative of that place where they have been for hundreds of years. That's the reason for the two-dimensional structure that's there. That's the reason these two guys are popping out. Okay, so we understand that, but we understand more because since we can work out the underlying bulk distribution under this model, we have a prediction for what the Scree plot should look like. We understand what Cattell did not. It shouldn't be a straight line. It was, obviously he did some hand interpolation of one example, and then that imputation that he did followed for 50 years. Everyone believes that, but what we understand now is no. It has something to do with the quantiles of the Marchenko-Pasteur law, and it is what it is. It's just some shape. As the shape factor goes closer to square, it becomes more curved. As the shape factor goes closer to tall, it becomes closer to a straight line. So if the case was something where you had many, many more rows than columns, Cattell's structure might become approximately correct. Now, as a statistician, what I would prefer to do is what's called a QQ plot, where what you do is you take the predicted ordered eigenvalues from an underlying distribution, Marchenko-Pasteur with a given shape factor, and you plot them against the observed eigenvalues, and those should be on the identity line if your theory is any good. So when we do this on the genes from geography data set, this is the identity line, and you see that there's an excellent match, except for divergence up here. However, I just want to make clear one thing. The Marchenko-Pasteur that we used is not the one you get from the straightforward shape factor. We had to make an adjustment because there's something in the data that's called linkage disequilibrium that means that the apparent shape factor is not really the right one. And there's a paper by Nick Patterson and Alkis Price that explains this in some detail if you care. So if you try to do this, you have to use a different beta than what you might think. Okay, now another thing that we saw is we know that the eigenvectors have a certain cosine between the true population eigenvector and the recovered one that's present. So, and in particular, we know that that depends on the strength of the underlying corresponding eigenvalue. And so stronger eigenvalues lead to smaller cosine, to larger cosines or smaller angles, and then smaller eigenvalues lead to more discrepancy between those, right? So what that has to do with is the PC1 has a much larger eigenvalue than PC2. So what you would expect to come out of this is that sort of the location probable ellipse would have structure like this. If I'm trying to tie down an individual from their principal component scores, well, it'll be much more accurate in the PC1 direction than it will be in the PC2 direction. And once you understand that that's present in the expressions that I've already told you, now look at Italy. Okay, we know on the map that Italy has this boot structure and it's very narrow at some places, but then at another place, when you look at its reconstruction, it's reconstructed much more broadly here. but that width is in the direction of PC2. So you again see the spiked model at work. It explains the phenomena that you see in this plot. So we're pretty good at determining where you are in latitude, not so good at determining where you are in longitude simply from genetic data. Now, maybe there's other explanations for this. Maybe people move around more in longitude than they do in latitude. Certainly, the spread of agriculture in human populations was that way, because if you have crops at a given fixed latitude, you're more likely to be able to grow those same crops by staying at that latitude and not migrating north and south. So it's not only the mathematics of the spike model, there could also be some influence of human behavioral choices. Are there any questions? Okay, so we'll make a couple applications of this. So how am I doing for time?
18 minutes.
Okay, so anyway, I've introduced to you some material about the spiked model, and I hope I've emphasized that all of this was not the work of the authors. My co-author Ian introduced the terminology spiked model. But what we're doing in the work I'm about to describe is use the spiked model for various things that statisticians think are worthwhile applications of it. So just, again, illustrate more uses of the spiked model. And because of time, I may abbreviate some of the explanations, but I think I've mentioned that there are papers that you may consult. I'd be happy to talk with you offline. So a matrix denoising problem would be something where you have a low rank structure plus noise, and then you have this object which has some hidden low rank structure that you want to recover. And the particular technique that we're going to talk about is singular value shrinkage, where what we do is we take the singular values of the matrix, which matrix Y, which I'll call little yi. They're attached to the observed matrix capital Y, and we apply some nonlinearity, which could be simply truncation. And then out of that, we get adjusted singular values, and then we rebuild the matrix. And this is an idea that's been around a long time in various guises. Now, the point that I want to make is there are some things that come out of the spiked model that provide some insights that you might want to know. I would say some pioneers in pointing out the importance of the spike model for this. Andrew Nobel and Andrei Shabalin, I would rank up there, and you can see there are many other approaches to this problem. There's papers by Nodler and Critchman for a kind of optimal test of significance of eigenvalues, and the literature on this extends into hundreds of papers. I've just mentioned different approaches here, many different approaches. The closest to what we're discussing is this one here. So here's a simple example that we claim is a corollary of all of those results on the spiked model. It says, if I have a square matrix and I have white noise added into a low rank structure, then here's what I ought to do. Truncate the singular values at a specific place. It's just a number, and if we had the problem scaled appropriately, then we could just say that number is 4 over square root of 3. So if the singular value is larger than 4 over square root of 3, keep. If it's smaller, kill, and rebuild the matrix. But you may be willing to do something more sophisticated, and if you have dreams of doing something that is not as brutal as keep and kill, but you kind of want to massage the singular values rather than destroy them, well, then this is the best massaging that you can do. And asymptotically, it's like leaving things alone. If they're very big, you don't touch them. But if they get close to the edge of the quarter circle law, which would be at 2, then they're going to be very small, and at 2 they actually vanish. So if you don't know the noise level, then you take the median singular value, and then there's a specific conversion, and that gives you your estimate of the singular. So this is a simple rule. We claim this is just a straightforward corollary of the spiked model. And the way that you work it out is ask yourself, if you decided to truncate the singular values at a given value tau, under the asymptotic model we're talking about, say the matrix is square and we scale things so the noise level is 1 over root n, then it turns out that for the singular values that are below the threshold that you've chosen, well, you're not going to be estimating them, so you're going to suffer this loss. On the other hand, for the singular values that are larger than this, you're going to suffer a loss that has something to do with the signal strength, but also has this sort of basic estimation noise factor of 2 built in. And if you study that expression, it'll immediately become clear that tau equals 4 over square root of 3 is it. Like you really shouldn't consider any other tau, because that tau is the only one that makes sense. There is no other threshold to be used in this particular case. And if we consider examples of different problem sizes as we go through that, and we have different noise levels as we go, different noise types as we go this way, so Gaussian, uniform, student, 20 by 20, 50 by 50, 100 by 100 with different matrices. What people would do using the Scree plot is look for the elbow and truncate. That's the red line. And what we recommend is the blue line. And you can see that the blue is better than the red substantially. And the key point is down here in the regime where the truncating at the edge of the spectrum would seem to be the right thing to do. There's a very significant amount of mean squared error that you're going to suffer. And the reason is that the singular vectors are just so noisy at that point that it's not worth using them. So basically, there's some place at which the singular values correlate with the phenomenon you're interested in, but the correlation is too weak for you to make productive use of them if you're just going to truncate. This can be elaborated. You can write code. It's very easy to implement. So the spike model tells you there's a very simple answer to this, where for hundreds of papers, people have dreamed up dozens and dozens of approaches. This is what the spike model says, and it just says there is no other answer when the spike model holds. And you can make comparisons between the optimal shrinker, optimal truncation, and some of the other things that people have tried, such as bulk edge thresholding or soft thresholding. And as a function of rank, you can see at each given rank you'll do better by using these approaches, which is necessary since they're optimal. As far as shrinkage, we've mentioned the optimal rule for Frobenius loss, and you can say, well, why should I care about Frobenius? If you chose operator norm loss, there's a different nonlinearity. If you want nuclear norm loss, which is the sum of singular value discrepancies, then there's another one. And so here, this means debiasing of singular values. That is, we've seen there's a bias. X of y is the nonlinearity that undoes that bias. So that turns out to be the optimal policy when you're in the face of operator norm loss. And it's a very clear expression. As a function of the shape factor beta, these nonlinearities can be plotted. I guess there should be a green, a red, and blue curve, but it doesn't project very well with these color guns here. In any event, you can come up with the correct nonlinearities. Okay, how much time? So we don't have that much time. What I'm going to do is skip covariance, just ignore everything I say. But somehow this extends to covariance, and there's very similar stuff of what's the best way to estimate a covariance, and the paper's out there. Now, I just want to point out that what we've done so far is discuss what happens if we have a very particular noise model, which might be realistic for, say, radio frequency signal processing. It might seem crazy that you could use it in the genomics case that I mentioned, and yet we've seen that it kind of works with some adjustments. There's the problems we've considered where this noise comes in with rank 1 structure. There's the covariance modeling we didn't discuss, where there's also a factor structure of similar nature. In everything so far, the bulk of the empirical eigenvalues was Marchenko-Pastur. Now, actually, scientifically, this is a special case, and we ought to be ready to model our data much more flexibly. Unfortunately, there's not that much time in this talk to go through this in detail. I'll just convince you this needs to be done and mention to you that you can now do it. So let's go over a little bit of that. In a more flexible case, you'd have some other kinds of noise models, and you'd get some other kind of bulk. So if you go in computational biology, you don't always see Marchenko-Pastur. And so there's an example that we can talk about in a paper from Brick and Silverstein using International HapMap Consortium data where Marchenko-Pastur does not work. There's a paper of Bouchot and Potters in Empirical Finance where they show the Marchenko-Pastur is here and it doesn't match what you see in the actual data. The Marchenko-Pastur story is a nice one, a towering achievement of random matrix theory, but for empirical work, we would like to be able to go beyond that. In the Scree plot, that example that I just mentioned with that Marchenko-Pastur is not quite right. In the Scree plot, if we only had ever gotten there, we might not understand this. When we go to QQ plots, we can actually look at the deviation of the empirical curve from the predicted theoretical curve. And when we do that, we start to see significant deviations of order 1. And so we can start to understand that Marchenko-Pastur won't do everything for us. We need a more flexible modeling approach. Some examples of models that could come up, examples of models that could come up, you could have colored noise. You could have white noise, but in the rows, you collect the data with exponential decay so that you forget data from long ago with a time constant on your memory. You could have heteroscedastic rows. You could have time series dependency in either rows or columns. These are all examples that would push you away from Marchenko-Pastur, and yet we can easily think of modeling situations where these would come up. Now, what's been going on in the talk to date was that the underlying distribution of the eigenvalues of the data was concentrated because we had an identity matrix for the covariance of the data. And this spike at 1.0 was broadened by the particular process of getting empirical eigenvalues into the full Marchenko-Pastur bulk. That's called the Marchenko-Pastur mapping that takes the underlying story and broadens it out. Now, if we had a different structure, the Marchenko-Pastur mapping would give us other correspondences. So if, for example, the eigenvalues could be one of 10 linearly spaced things, then we would get something like this for the bulk distribution. Okay, so there's a complicated nonlinear mapping that does this. It's not a straightforward convolution, but it's associated with what in random matrix theory is called free convolution. In the autoregressive story that I mentioned, the population spectral distribution has a certain shape that in this particular example at about 5 3/4, but what you get under the Marchenko-Pastur mapping will get the eigenvalues to spread out to about 8. So you get differences in shape. Now, for a long time, people have asserted that you can make computations of the Marchenko-Pastur mapping by using a so-called fixed point relationship that was known to Marchenko-Pastur and in a different form it has been used productively by Jack Silverstein. But actually, so Edgar, so he's a graduate student at Stanford, Edgar Doberman, and I gave him this problem of do something better than the fixed point thing. And he really succeeded and has done an excellent piece of work, which is now in random matrix theory and applications. His software is called Spectrode. It computed all the examples that we just saw by a new principle, and it's truly of high accuracy and allows you to do precise computations of the Marchenko-Pasteur mapping, where in the past people kind of waved their hands. Once you can do these precise computations, all sorts of new options are available to you as a statistician because accurate empirical modeling becomes possible. So I don't have time to go into detail of how we would use this, but I'll simply say it's now possible to understand how we can replace the arrays Zij, which were present in the talk before this, involving white Gaussian noise, by other situations. We can calculate the underlying bulk, and not only the bulk, we can calculate all the parameters of the spiked phenomena. So in this general case, in some abstract way, the structure we've talked about continues to exist. The eigenvalue biasing can be calculated. The cosine displacement of the eigenvectors can be calculated. These are all definite quantities that continue to exist in this more general setting, and you can calculate them stably with software, so on and so forth. Now, importantly, the optimal procedures that we've spoken about, both in the denoising case and in the covariance estimation, just depend on these spike descriptors. They don't depend on other things. So they've actually already been worked out. You simply plug in the spike descriptors. And so I'll skip. And some of the math that should be mentioned about this, of why this all works and can be generalized beyond the white noise case, I would mention the papers of Benesh George and Rajrao Nada Kuditi, or Rajrao Nada Kuditi's nice paper on shrinkage. You can also see the book of Yao, Zheng, and Bai. A simple example is that we can now take that 4 over square root of 3 principle for truncation of singular values in the case where we have Marchenko-Pasteur bulk. And we can say, more generally, there's a functional, call it T of f, which has to do with you plug in the bulk associated with the observed data, and it will tell you where you truncate. And you can write out explicitly what it is in terms of Stilches transform-like quantities, and you can prove various well-posedness and continuity and Fréchet differentiability properties of this functional, which therefore imply that all of these things are estimable at very reasonable rates, and you can get accurate estimates of them from empirical data simply by taking the underlying bulk distribution, cutting away the top eigenvalues, and filling in either by zeros or by something that's imputed, which is described in the paper. Then simply apply this functional. It tells you where to cut, and just like 4 over square root of 3, it achieves for you the optimal asymptotic mean squared error when you do it, so it's really the right place to cut. So coming out of this more general approach, we learn how to truncate the singular values and eigenvalues even when the Marchenko-Pasteur setup is not the precise correct one, but where some other underlying bulk shape is present. So I'm out of time. I want to thank you for your attention, and again, thanks to the Feder family for this wonderful occasion.
Questions? So all those results are asymptotic. So how do you know if 1,000 is close enough to infinity to use them?
So the question is, how do we know the asymptotic theory works? Essentially, the way that it works is that what matters is the total number of elements in the matrix. So for a matrix that's thousands by hundreds of thousands, there's a huge number of elements in the matrix. And roughly speaking, the discrepancy between the asymptotic theory and the finite sample has to do with the difference between the empirical CDFs that are involved and the limiting CDF. And that goes like 1 over the number of matrix elements. So not some other power, but 1 over. So that helps quite a bit. I would say that's the dominant thing. So if we were down at a smallish size, like 10 by 10, you might think that this approach is not so good. Even in that case, it's been checked pretty carefully, and it may be just fortuitously in the examples people check. Maybe there's some things they could have checked where it doesn't work. I don't know, but it seems to converge pretty rapidly. The lack of applicability of theory, there may be situations where the theory we talk about is not applicable. I don't think it's the matrix size problem, but there could be other things wrong with the matrix that put artifacts and mess things up. So if you're looking for problems in this theory, so here would be an example. In the genomics data, if you happen to have related people, so parents and children or twins, things like that, and if they happen with appreciable frequency in the population, then that changes the bulk shape in a noticeable way. And so it's important to collect the data correctly in order to make some of this theory be correct. Another example is there's something called linkage disequilibrium, which has to do with SNPs that occur too close together on the genome. They're so highly correlated, they also cause disruptions in this theory. There are data that mess this all up. It's not really an end-dependent thing, it's more like a reality-dependent thing.
In the mapping you showed with the genomic, you showed the 2 PCA components. And once you realize that they are implied by the geography, the manifold on the plane, and you know that the eigenvalues are different, if you normalize by the inverse of the singular values, that should, why haven't you done that so that the plot will have a circular Iso surfaces rather than ellipsoid Iso surfaces?
You're a real statistician. Okay, statisticians always like to put things in coordinates where they're natural from the point of view of the standard error, like they're the same amount of error in every direction. But it's inherent here that there's more uncertainty about the geographic coordinate in one direction than another. It's exactly visible in the spike model. So if we measure error in kilometers, we can't solve that. If we measure error in standard deviations, then we can do what you say. But most people are not statisticians and don't like to measure in standard deviations. They prefer kilometers.
Another question. If the matrix you displayed on the genomic data has a rectangular structure which is 100 times larger in one dimension, in this case, wouldn't it be true that the singular values will converge to a constant? Because the noise, let's disregard the genomic data, just take the normal values. So they're uncorrelated, and the ground matrix should be identical. So all the eigenvalues should be the same, should not comply to any curve.
So you're basically saying if beta is 1 over 100, then basically the Marchenko-Pasteur should collapse to a delta function at 1.0. That's another way to what you just said, right? So actually the beta that we're using is not 1 over 100 because as I mentioned, there's this problem of linkage disequilibrium. You can measure lots and lots of SNPs, and you can think that you're getting 600,000 independent SNPs this way. But the effective number of independent columns is much smaller. And there's a way, in the paper of Nick Patterson, Elkis, Price, and David Reich in Nature Genomics, 2006, they explain a way to calculate the effective number of SNPs. So what we used was a beta of about, I think it's closer to 0.13 rather than 1/100. And that indicates, so at 0.13, there's still significant broadening. It doesn't collapse to a point. But it's an excellent question.
Last question.
So talking about the eigenvectors, if you are interested in recovering the spike, is PCA the best you can do, or can you go to higher order statistics to do better?
Well, if you know something about the eigenvector, then you can use that, right? So presumably, you could do an even better job of recovering the underlying structure by using the map of Europe, right? But and so, right? I mean, we could somehow iterate, right? We could use the map of Europe, and we could somehow know where people are from, and we could say, well, this person ought to be from Geneva, but he's being located in Neuchâtel. Let's move him over, right? We could do stuff like that.
Not what I'm suggesting at all.
Okay.
What I'm suggesting is simply not to use PCA. PCA is just one method for analyzing the data. You can use other moments, for instance.
There are many different estimation principles you can bring to bear if you have other kinds of information than what I've discussed. You're totally right. And this is just a workhorse of science and technology precisely because it's so easy and assumes so little. So I don't want to say this is the, asymptotically most perfect thing to do, because in a real scientific setting, you may know other things. You're totally correct.
Let's thank the speaker again.